All right, so we're going to start talking about the functions of nerves, how nerves work, and just general function of the nervous system. This will probably take 2 lectures, maybe even a third, just depending on how fast we're able to go through it. And so, just first, let's talk about this concept of membrane potential. Now, you know that the mousetrap is perhaps one of my favorite illustrations, just because, I mean, look at it; it's perfect. It has so much going for it. Um, So cells, all cells, have this thing called membrane potential, and this just means that so you have a cell membrane, and along that membrane there is the potential to do work. You see this mousetrap; it has the potential to do something, right? Well, most cells, that potential kind of is steady. The cell maintains a certain potential. In nerve cells, that potential can rapidly change. And so nerve cells are highly excitable, is what we say about them. That they are, when they change, that change can produce a big output of energy. And that energy we know is electrical energy. So as we talk about electricity, let's talk about some basic principles of electricity. And I think we all understand these opposite charges are attracted to each other. The positives in the circle are attracted to the minuses or the negatives in the rectangle there. They really want to be with each other. And so, in order to keep them apart, energy is required, right? And so, if that circle and that rectangle really want to be together, it takes a little bit of energy to keep them separate from one another. And so, that energy is that potential to do something, right? And when those two forces, when that circle and that rectangle move closer to each other, that energy is then released. All right. So this is kind of how a nerve cell works. You have one side of the membrane is attracted to the other side of the membrane. And when those two things meet, that energy can be released. And so that potential in that membrane has the potential to do work. And we call this, on a nerve cell, we call this the resting membrane potential. So go back to my mousetrap. That mousetrap is currently at rest. However, it is loaded. It's ready to do something. It's as soon as it gets triggered, it is going to do the thing. All right. That mouse trapper part really wants to slam down against that wood. All right. But it's currently at rest. It just needs a mouse to come by and eat whatever that thing is. All right. And then it will do that work. All right. So how does this work in a nerve cell? Well, here's a cell membrane, and here's the intracellular composition and the extracellular outside of the cell composition. So on the inside of the cell, or let's start at the outside of the cell. On the outside of the cell, you have sodium ions, you have chlorine ions, all right, and you have a little bit of a potassium, but not very much potassium at all on the outside. And so we say that the outside of the cell is slightly positive. It has a slightly positive charge to it. The inside of the cell has all this potassium in there, not very much sodium, not very much chlorine, but it also has these A's stand for just really big proteins that are inside that cell. And those proteins are causing the inside of the cell to be slightly negative. All right. And so you have this slightly positive on one side and slightly negative on the other side. And again, remember, they're attracted to each other. And so the cell is maintaining this, what is called this resting potential. It's at rest, but it has the potential to do some work. All right. Now, one of the ways that it's maintained, and I don't have a picture of this, sadly, it's probably on one of these pictures here. I'll see if I can find it. Nope, of course I don't. All right. So one of the ways that this is maintained, and we'll just use this here, there are these structures on the membrane called sodium potassium pumps. And guess what they do? They pump sodium and potassium. And so sodium is pumped out of the cell and potassium is pumped into the cell. And these pumps maintain this resting potential. Sodium pumped into, potassium pumped or sodium pumped out of potassium pumped into the cell, and they maintain this resting potential. They maintain this cell's ability to do work. All right. Now, eventually, that cell can be triggered, and it can cause one of two responses. It can cause a graded potential or an action potential. And so let's talk about, first of all, let's just talk about what can trigger these things. So think of a game of Jenga, right? And you're building, or you build this tower, and then slowly you take away pieces of that tower. And as you take away pieces, that tower has the potential to fall over, right? It always has the potential to fall over, but you are creating a greater potential to do that. A graded potential is when it almost falls over. There's not quite enough stimulus to get it to go all the way, right? It's graded, meaning that it's not 100%. It is just kind of toward toward the top, but not all the way. This could be some sort of stimulus that's from the outside world. It could be sensory stimulus that is causing this graded potential. All right, so it's not 100% of that. These are usually short-lived and localized, and so they don't go all the way. The stronger the stimulus, the more change there is, and the further that current flows. So the stronger the stimulus, and you think about the Jenga game, the stronger the stimulus, the more you pull on it, the more it might go over, but it doesn't go all the way over, right? That's different than an action potential. An action potential is an all the way kind of thing. This is, if you want to go back to the mousetrap, even a certain kind of mouse could sit on that and eat and not trigger that. But an action potential is when that little lever gets stepped on, there's no halfway to that. When that mousetrap actually fires, there's no halfway to it. It goes all the way. And so an action potential is an all the way firing of a nerve. And this is the principal way that nerves send signals. It's a long distance kind of communication. You'll typically see this on the axon of a neuron. And a lot of times the action potential is referred to as the nerve impulse. All right. And so again, you're typically going to have one of two kinds of input that cause these. It could be sensory input from the outside, or it's going to be the actual nerve impulse from another nerve, or the chemical signal from another nerve that's going to cause one of these kinds of potentials. We won't spend very much time talking about graded potential. We're going to spend most of our time talking about the action potential, which is what we're getting ready to go into a little bit more. How is an action potential generated? It's basically 4 steps. You can kind of go from left to right here with a little bit of caveat between the two. And so the first step is you're at resting, right? All of the gates are closed. You can see here these channels here. Oh, and here's the sodium potassium pump doing the thing, right? Potassium out, potassium in, sodium out. Good. All right, keeping it all in order. All right. And so this is resting potential. These cells, or these ions, really want to go in, and these ions really want to go out, but they're being kept from that. All right, so that's resting potential. If something triggers this nerve to where it is no longer able to keep those charges separated, that is called depolarization. It could be the nerve impulse traveling down the length of the nerve. It could be sensory input that triggers this nerve, but it depolarizes it. What does that mean? Well, the first thing that's going to happen is that sodium is going to rush into the cell. And so right now, this has a positive charge, out in here has a negative charge. When sodium goes in, the outside is going to be negative, as you see here, and the inside is going to be positive. It has depolarized it. It has completely switched those poles. And this depolarization causes the action potential to occur. I like to think of it as a wave. Like if you're at a stadium and there's people there and they're doing the wave. All right. it takes a while for that wave to get started, but once it reaches a certain threshold, the wave will actually get going around the stadium. All right. And so it's the same way here. That sodium starts filling the inside of the cell, and once it reaches a certain threshold, it will fire the action potential. And you go back to our mousetrap. That lever takes a certain amount of weight to cause that lever to fire, but once it fires, it's all the way firing. All right, and so that is what we call the threshold. All right, it takes a certain amount to happen. Well, that's depolarization, step 2. So the action potential has fired and it's traveling down the nerve now. Well, then the cell will repolarize itself, step 3. So repolarization, potassium will go out of the cell to repolarize the cell. Notice it's repolarized. Got the plus out here on the outside, the minus here on the inside again. That's good, but it's not back to where it should be. It's not reset. And so the next step that will happen is the sodium is going to be pumped out and the potassium is going to be pumped in. Now, what will happen during this step is something called hyperpolarization. And hyperpolarization, the best way to think about it is this is when the membrane is returned back to resting potential, but a little bit too much. And it returns it just a little bit too much. It allows for that resting potential to be perfectly fine-tuned to get it back to normal. All right. And so after repolarization, there's something called hyperpolarization, and the hyperpolarization settles back out and returns the nerve back to resting potential. And so I know that takes a while to explain this, but this is happening almost instantaneously. The way that you can easily think about this is sodium and potassium are just basically switching places over and over again in order to trigger that action potential. Now, how does this go down the length of an axon? Well, we call this, when that signal travels down the length of an axon, we call this propagation. It's spreading. We think of the word propaganda as to spread information. And so this is spreading, but it's going all the way down the length of the axon. All right. And so each time when depolarization happens in one section, it triggers depolarization in the next section, and repolarization is happening back behind it. And so you have this constant cycling. All right. It can happen in one of two ways. In an axon that doesn't have the myelin sheath on it, it's just this continuous conduction down the length of the axon. As you see here, continuous conduction. Where there are myelin sheath, remember these gaps we talked about in the previous video, and here it calls them nodes of Ranvier. They're not called that anymore, I don't think, but it's an okay term to use. Basically, there's just a small section of exposed axon that's not covered by this nonpolar substance. And so what happens is the action potential will actually jump over those sections of myelin sheath. And so it's skipping large portions. I mean, think about it this way. If I gave you a 200-page book and I said, read the whole thing, oh yeah, and you only have to read one page in every 10. So basically you're only reading 20 pages. You're going to read through that book really quick if you only have to read 20 pages. And so this type of conduction happens really quickly. And that's why when you see something, or like say you touch something that's hot, you're able to jerk your hand back really quickly. That happened very fast because of this kind of saltatory conduction. Why is it called saltatory conduction? It has nothing to do with salt. It comes from the Latin word saltare, which means to jump. And so what's happening is the action potential is literally jumping over these sections of myelin sheath. And that's how the action potential is propagated. Now, during and after, right after an action potential, there's a period called the refractory period. And a refractory period is just this. It is a time when another action potential cannot be triggered. It makes sense. I mean, if you want to go back all the way back to, well, we can use Jingo, we can use Mousetrap, either way. This Mousetrap is just fired, or it's in the middle of firing, that is called a refractory period. There's no way that can be reset right then. It takes some time, just like it takes some time to set this business back up. All right. Once it falls over, you can't fix it in the middle of falling. That's called a refractory period. Also, it takes some time to set it back up, get it back to normal. And so that period of time is very small, as you can see here. This is milliseconds. All right. There is a small amount of refractory period that is required to reset the nerve. And then lastly, let's talk about some imbalances that can occur. The big one here, talking about the myelin sheath, there's a disease called multiple sclerosis. Sclerosis is, multiple, more than one. Sclerosis. A sclerosis is basically where you have a section of your myelin sheath that has been damaged. And this particular disorder is an autoimmune disorder, meaning the body itself is attacking the myelin sheath and attacking the Schwann cells that create it. And it creates these, basically these lesions on the nerve that are called sclerosis or scleroses. And the nerve doesn't able to work as well. The conduction slows in those sections, and so it can cause pain because there's no longer protection of the outside tissues are no longer receiving protection, and it can also cause slowing of movement. It could lead to some very serious problems because if conduction ceases altogether, it may lead to something like paralysis. And so this is an example of an autoimmune disease, meaning this is when the body is attacking its own tissues. And so that's multiple sclerosis.